Everyone, we'll get started in about 30 seconds. All right. Great. So, hi everybody. Thanks for coming. I'm so pleased to introduce our final speaker today of 2020, Dr. Melissa Coleman, who's our last speaker of the year and is therefore the headlining act of the 2020 Systems Neuroethology Seminar Series. Dr. Melissa Coleman is a professor at Claremont McKenna College in California. Her contributions to neuroscience are as broad as they are impressive. Mel received her PhD from the University of Alabama at Birmingham, where she worked with Michael Nussbaum, studying the mechanisms of synaptic transmission and modulation in the crab's stomatogastric ganglion. And I can't help but interject to share one of my favorite science tidbits. For those familiar with the STG, the MCN1 neuron is actually named after our esteemed speaker and is the Melissa Coleman neuron. After her PhD, she then went on to work with Richard Mooney on birdsong in the zebrafinch. There, she made important synaptic and circuit-level insights into the song system, uncovering, for example, how a thalamic nucleus can connect auditory brainstem circuitry to the HVC, which is one of the key nodes in the song system, and moreover, how these thalamic inputs can actually modulate HVC activity. In her own group, Mel's continued to work on synaptic and network properties underlying birdsong, with work in zebrafinches ranging from detailed synaptic characterization to the effects of important modulators like estrogen and dopamine on song preference in both males and in the understudied females. Mel has also been an important part of the Grass Fellowship programs at Woods Hole. She was a fellow in the late 90s and has since been a trustee, and more recently the director of this fantastic program, which brings grad students and postdocs to Woods Hole to pursue exciting, independent, and most importantly, funded research programs for the summer. I met Mel at Woods Hole and am always impressed with how she mixes deep scientific insights, kindness, and critical feedback. I think this really reflects her outstanding teaching skills and the care she takes in educating the next generation of neuroscientists, including myself. I always leave a conversation with Mel with something new and challenging and fun to think about. Today, she'll talk about one of my favorite projects, studying the neural basis of a gorgeous natural behavior, the duetting song of Duetting Wrens. Thanks, Mel, for joining us today, and I can't wait to hear all of what you have to tell us.
All right, thank you, Emily, for that really fantastic introduction. I hope I live up to a part of that today. It's really a pleasure and an honor to be here today and tell you about this beautiful story and our lovely birds, the plain-tailed wrens. And so what I'm interested in is how, as everyone else, how the nervous system produces behaviors. And the behavior I'm going to talk about is a coordination of behaviors between two animals, and specifically focusing on these duetting wrens. And so in this picture, these are our subjects. In the picture is the male and female wren, and they produce a beautiful behavior. And so why do we care about these things? As a neuroethologist, we think of the most simple behavior, where an animal receives a stimulus and that affects the nervous system, activates some circuitry, and produces a behavior. And in the simplest case, this could be a pattern generating circuit. And the classic example is of egg retrieval in the goose, where the egg is the stimulus, and then the animal produces a fixed action pattern that retrieves the egg and puts it under her back to the nest. But of course, this is a very simplistic view of things, and as the title of the series is Systems Neuroecology, most animals don't operate by themselves, but operate together. And so what we're interested in is interactions between two animals, and how does the nervous system coordinate activity between two individuals. And so in this case, we have one animal that acts as a sender that sends a message to the other animal who is the receiver, and has some response. And this is a dynamic interaction between these two animals. And so I've just listed a couple of sort of classic examples here, where you have the blue-footed booby, where the male is showing off his beautiful feet to the female. He's the sender, and the female is the receiver of this information, and is making some judgments about the male and how beautiful his blue feet are. But she's also then sending a signal back to the male, and he's responding appropriately to that signal, or maybe inappropriately to that signal. We also have example of cichlids, who the male on the left is being quite aggressive to the male on the right. And so one animal is the sender, the other animal is the receiver, but the receiver is also taking that information and sending signals back to the other animal who's interacting. And so how is the nervous system coordinating the activity between these two individuals? Another really important example of this is in vocal communication. And so here, the final example are two people talking with each other. And in order to communicate effectively, one person is the sender and one is the receiver. And you have to have a nice alternation between the activity of these two individuals, between the speech patterns of these two individuals, turn taking, in order to have this conversation. So how is it that the nervous system coordinates activity across individuals? And so we're studying this in this beautiful example of the plain-tailed wrens that sing this really amazing duet. And so I've already introduced these to you. So the females on the top and the males on the bottom. And now I'll play you the behavior that we're studying is this duet. So that song is actually sung by two animals in a very tight coordination. And in order to show that to you, I'm going to show you a video of, this is a different pair, a video of two birds singing. On the top is the male, on the bottom is the female, and then across the bottom is a sonogram showing, which we've color-coded. The female parts are in pink and the male parts are in blue. And I'll play that video for you now. So what you can hopefully see and probably can't hear is that song is actually a very tight coordination between the female producing her part and the male producing his part, and going back and forth and back and forth. And because it's such an extraordinary behavior, I'll show you that to you once again. And so what we want to understand is how the two brains of these individuals can coordinate to produce this really fantastic behavior. And before I go on, I want to introduce, say that this isn't work of my own, but this is a long time collaboration with Eric Fortune, who's at the New Jersey Institute of Technology. And this has really been just one of those really fun experiments that you get to do for a long time. Here we are in Ecuador together. And then recently we had the great idea to bring along Nancy Day, who's helped us with a lot of the physiology, who was at UCLA with Stephanie White, and now has her own lab at Whitman College. And we also had a bunch of undergraduates helping with us from the Claremont colleges, including Hannah and Karun and Leanne, and also helped by Carlos Rodriguez, Ecuadorian student who's now a postdoc at UT Austin. So all these birds actually live on the slopes of the Andes in Ecuador, and this next image is just their habitat. So here's Ecuador outlined here. Can you see my pointer? Okay. The Ecuador is here, and then here is Peru. And these are the Andes, and all the little purple spots are where these birds are found. And they live in this thick, dense Chusquea bamboo. And here's Eric and Carlos looking for our duetting wrens. And how we find them, obviously it's very hard to see them. Eric tells a story of when he was first looking for them. He asked a well-known bird guide about these birds, and he says, oh yes, I know them, they're yellow. They're not yellow, as you've seen, and it's because they live in this dense bamboo, they're actually hard to find. And so how we find them is we play back songs to them, and then they approach our speaker. And so here's Eric and Carlos looking for our birds. So all the experiments I'll be telling you about were done in Ecuador. We're not allowed to export the birds, and I don't think they would do very well if we did that. Most of the experiments were done at the Yanayacu Biological Field Station, which is shown here. And so we do our surgeries in this little room, and most of the experiments were done up over here, and the kitchen's down here. And then our living quarters on the backside. One of the great things about field work, at least this field work, is we can use machetes to hack our way through bamboo. And so here's Eric and I going to find our wrens, going through this very thick bamboo. And then here's an image of some of the scenery we have, so it doesn't suck. All right, so just to remind you, here's the behavior that we're talking about, and this is really from a paper that started this whole thing. Eric and I actually were in at Woods Hole in Neural Systems Behavior course and started talking about this, which started the collaboration. But here's the image, and so here's a duet between these between two individuals, showing frequency over time. And this duet has 4 motifs where the two animals sing A, B, C, D, A, B, C, D, A, B, C, D, and it's an alternation. So the male is singing A and C, and the female singing B and D. And I'll play that for you just so you can hear that. And so what we're interested in is how the brains of the two individuals are able to produce this coordinated activity. And what I'll be telling you about today is dividing my talk into 2 main parts. First is the behavior, and I'm going to show you some evidence that it's actually auditory feedback from one individual to the other one that's the critical sensory signal from the sender to the receiver. And then the second part of that is actually getting into the brain and then looking at coding of signals in the sender and the receiver. Okay, so we can get clues about how this happens in the brains of individuals just by looking at the behavior. And so we know that there has to be the animal has to be able to produce the behavior. So there's some premotor activity that will occur. Then that animal will produce an auditory signal that's sent to the receiver and alter and influence the timing of the behavior in the second individual. And so we've come up with a basic schematic to show this. And so here on the top is a sonogram, a spectrogram. And here's activity in the male brain and activity in the female brain. And so when the male sings, that syllable is produced by his motor system. And we think there's a central pattern generating circuit involved based on work from other songbird species. That syllable then becomes the auditory, becomes the sensory signal that influences the auditory system in the female, that influences the timing of her motor system. She then produces a syllable that is sent to the female, sent to the male, excuse me, is received by the male, and that influences the motor pattern system in the male, and then he produces his syllable. And so there's this back and forth, and the linkages between the two animals is via the sensory input from one to the other one. So first I'm going to talk about the behavior and show you a couple of lines of evidence to suggest that it's this auditory feedback that's the critical signal from the sender to the receiver. And the first clues that we have of this are actually from recordings in the field where the birds were just singing and there was no manipulation. And so this is I was going to say experiment, but it's not, it's just birds behaving. On the top are 2 birds in a duet, and on the bottom is the female singing by herself. And what we find is that when the female receives input from the male, the timing of her syllables changes. And so I'll just go through this slide to show you that. So on the top is the duet, and the duet, the female singing ABD, 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 and the male is interspersed with CE, CE, CE. And then on the top, what we're showing is the timing, the intersyllable interval between the female syllables. So the timing of the interval between A and B, which is in green, between B and D, which is in orange, and between D and A, which is in yellow. And so I'll play that song for you now. Okay, so it's a nice alternation between the two animals. And then in the second instance, the female was just singing alone, and I'll play that for you. And what we found with this is when the female sings alone, she sings the same syllables in the same sequence, but the timing in between the syllables changes, showing us that it's really the input from the partner that is influencing the timing of those syllables. And so what we've done to show that here is now we have syllable ABD, ABD. But when you look at the intersyllable interval, you see that in one case, the intersyllable interval has decreased. In the other case, the intersyllable interval has increased. And so feedback, again, feedback from the partner is changing the timing of when those syllables are produced. And so this also gives us a clue that perhaps this series of syllables in the female is produced by a pattern generating circuit in the brain of each individual, at least in the brain of the female in this case. We see this change in timing between syllables even on a very short time scale. So here's this great recording where we have the duet occurring, and so the spectrograms on the top and the sonograms on the bottom where we color-coded who's singing what. And at the top you see a very nice alternation between male and female singing the part. In the middle, the male, dropped out. So you see there's no blue here. And when the male dropped out, the female continued, but the timing of the intersyllable intervals changed slightly. And in this case, the intersyllable interval between A and C decreased, and between E and A also decreased. And I'll play that for you, and you can hopefully hear in the middle that there's a little bit more of a gap where she's still continuing her pattern, but the timing in between the syllables has changed a little bit. Yeah. Okay. So the timing of the syllable production is influenced by input from the partner. And I'll show you pieces of it's not surprising, but I'll show you a couple of pieces of evidence that show that it's actually the auditory input from the partner that's important for the timing of those syllables. So the first experiment, I'm just going to show you the video because I think it's fantastic, is we captured pairs of birds, put them in cages, and then didn't allow them to see each other. And so we put a barrier in between the two animals, and then asked, could they still sing? And so that experiment is shown here. And of course, because we're out in the middle of nowhere, the barrier was an umbrella, which is shown here. So this is the big black thing in the middle is an umbrella. And then there's a bird here in this cage and a bird over here in this cage. And here's the experiment. So the two animals, although they couldn't see each other, were perfectly fine at singing a duet. And in fact, this was a little bit surprising to us, not completely, but a little bit surprising to us, because whenever we see them in the field and they start singing, they actually come together, which suggests that potentially visual information was important. But visual information might be important, but it's not critical for producing a duet. So the second experiment we did to test whether auditory feedback was critical was we manipulated the distance between the two animals, which changed the time it takes for the signal from one animal to get to the other animal. And so that's illustrated in the next couple of slides, where here's the experiment in which you separated the animals by, 7 meters. And we have the male down at the bottom and the female at the top. And in this case, in this picture, we also have the umbrella in between them, the visual barrier in between them. We also have a microphone next to the female and a microphone next to the male. And so the top spectrogram is the recording from the microphone next to the female, and the bottom is the same song with recorded from the microphone next to the male. And so in the top panel, the female syllables are much louder, higher in amplitude because they're closer to the female, and the male syllables are much lower in amplitude because he's much further away. And so this experiment also helped us figure out who's singing what, because of course with this tightly coordinated duet, it's really hard to tell who's singing what. For example, here you have this little bitty syllable down here that's very low amplitude, and that's produced by the male because it's much quieter in the microphone next to the female. Okay, so here's the experiment at 7 meters. So the female produces her part here, and then it travels, that signal travels to the male, and it's picked up by the microphone near the male, so it's lower in amplitude. And then the male hears that and responds back to the female. And so now there's a delay between when the female sang and when she hears the male response, and then she hears the male response, and then that travels back to the other animal, and it goes back and forth. And so what we can do now is measure these delays between when the female sang and when the male response came back to the female. So here the female sings, it goes to the male sings and comes back. And so that delay is mostly due to the signal going from the female to the male and back from the male back to the female. And so we can measure all of those delays, and we expect that if auditory delays are important for this production, that those delays would get longer and longer over time. And in fact, that's what we see. So here's the response of those, the quantification of those data. And so on the bottom is the distance between one bird and the other bird. We have them either in the same cage together or up to 10 meters apart. And this is the interstimulus interval. And here's for the delay with the male and the delay with the female. And we find that the slope is about 5 meters, milliseconds per meter, which is around what we'd expect for the speed of sound, to get from one animal to there and then back again. So this is pretty cool, because what we've done is we've separated the birds, and the birds are actually able to compensate for this change in distance, suggesting that it's really auditory cues that are important for coordinating the duets. Okay. So that's really the end of the behavior part. And we have, here's the song, the behavior that we have produced by 2 individuals. The male produces a syllable that travels, that auditory signal travels to the female. She hears that and sends a signal to her motor system, and then she produces a behavior and sends a signal to the male. Okay, so what we really want to know is if let's see, sorry. What we really want to know is if this is really occurring in the brain. And so we want to go in and look at the brain to see how this duet is being performed by each individual. And so the next part of this, I'm going to look at coding of signals in the sender and the receiver in the brains of each individuals. And so to do that, what we needed to do was record from the brains of each individual. And where do you do that? Well, what I've shown you is that we expect to have a premotor activity. So if I go back, sorry. We expect to have premotor, we need to record from a place where you have premotor activity. That is also probably a pattern generating circuit, and also that receives auditory information about what the bird is hearing. And the location for that seems to be, is probably where we started, at least, was this area in the bird brain called HVC. And why we chose HVC is not only because it's at the surface of the brain, which seems uniquely created for neurophysiologists, but also because it's part of a motor system. So we know that it's involved in singing, And so as part of the motor system. It also contains, as we know from other animals, including the zebra finch, where this is much more well characterized, it contains a pattern generating circuit for song production, and it receives auditory input. So this seems like the bee's knees of the places in the brain to look about how these two animals are coordinating their activity. And so what we wanted to do then was go and record from animals as they were singing duets. And so we would capture two birds and implant electrodes in their brain and have wireless transmitters on their head to record activity from their brains as they were duetting. And we made a couple of predictions. So here we have two birds where we're recording from the brains of each individual. And we made predictions about what we would see in the sender and the receiver. We first predicted there'd be an increase in premotor activity in the brain of the sender. And so here in this duet, we have the female that's producing premotor activity when she's singing, and the male, whose HBC, is producing premotor activity when he's singing. So you see this alternation of activity between the two animals. In the second prediction, we had, oops, I didn't know my computer would do that. There we go. In the second prediction we had is that there would be excitatory auditory responses in HVC of the receiver. And so because we know that auditory information is important for coordinating the activity between the two individuals, and in fact we know in other species, for example, in swamp sparrows, that you see an increase in HVC activity to playback of what the bird is singing in awake animals. And so we fully predicted that we would see, for example, when the female is singing, we would see premotor activity in the female, but that response, that auditory activity in the female would produce an auditory response in the male. They're doing construction outside my house on the sidewalk, and so I'm a little bit distracted, sorry. Of course. All right, so to do these experiments, we had to go record from the birds. And I just wanted to show pictures of our first laboratory because it's so spectacular. So these we did actually with urethane anesthetized animals, but here's the laboratory. It had a dirt floor, which was good because we had to have a separate ground that we could then feed into the ground. And this was our first setup. But thankfully, to the National Science Foundations, we were able to go back later with a little bit more information, with a little bit more money, and set up a lab now inside. And here's us actually setting up our lab. You see how much more sophisticated we are now, where we have suitcases for our table and a door that we got from down the road. We also have a heater for the animals to keep them warm during surgery. A nice microscope off to the side that we can do our surgery, our electronics here, manipulators, anesthesia machine, and then we keep going. And on the right here is Nancy Day making electrodes for our recordings. Okay, so what I'll show you in the next couple of videos are recordings, videos of these birds singing duets with the electrodes implanted on their head. So again, we implant electrodes on their head. These are wireless recordings. So we have the wireless electrode on the top of their head that gets sent to the electronics. So here's this experiment of that. The female is on the left and the male is on the right. We have food and water ad lib for them and bamboo in their cages. And I'll now play the video for you. So this is the male, and this is the little wireless transmitter on his head, and then we have a little backpack battery on the back of his head. And what I'll do on the next video is I'll show you this video again, but we've added the neural activity that we've recorded from the male and the female. So And now here's the male activity that you'll hear. And here's the female activity. All right, so impressively, I have to say, we're able to record neural activity from the brains of these animals as they're duetting. And so here's an example of another experiment, not the one I just showed you, but of another experiment in which what I'm showing here is the spectrogram of the animals as they were duetting. And so the female singing ABD, ABD, AB, and the male singing CE, CE, CE. And you get this nice alternation. And of course, at the same time, we're recording electrical activity from the HVC of the female. And we have actually the 4 wires in the brain. So this is electrode 1, 2, 3, and 4. So this is the action potential activity in each of the electrodes in HVC in the female. And then here is electrodes 1 through 4 in the brain of the male. So each line is the action potential in the brain of the male. And what you see, and we've color coded when the female sings in pink and when the male sings in blue. And so these are the individual spikes, and then we can create histograms based on those. And what we've done is just invert the histogram so you can see the timing in between the two. So here are the histograms of the female when she sings and the male when he sings. And so what we're asking is what happens in the brain of the sender. And we made this prediction that we would see premotor activity in the brain of the sender. And in fact, that's what we found. And so if you see here, when the female singing, you see increased activity in the female brain. And when the male singing, you see increased activity in the brain of the male, in HVC of the male. And that's quantified here on this next slide here, where we quantified the activity in HVC during syllable protection in the female and the male, and you see an increased activity in a premotor activity in the brain of the sender. And that's in fact what we predicted initially of what happens in the brain of the sender. We said that there would be an increase in premotor activity in HVC, and in fact, that's what we saw. The second prediction we made was, well, the second question we had was what happens in the brain of the receiver, and the prediction we made was that there would be excitatory auditory responses in the HVC of the receiver. And this is based on a lot of information, as I said before. And so if we go back and look at the recording I just showed you, and look to see what's happening in the brain of the receiver, you see that when the female is singing, there's not really much response in the brain of the male. And when the male is singing, there's not much response in the female. And so to quantify that, we just counted the number of spikes produced in the brain of the receiver compared to the baseline activity that occurred before they sang. And we did that. We found that there was actually a reduction in the number of action potentials in the brain of the receiver compared to baseline, suggesting that HVC is actually inhibited in the receiver. And, you know, in hindsight, that's not very surprising because you actually want 2 animals to alternate their activity. And if the premotor activity of one animal is inhibited while the other animal is singing, that would actually provide a nice neural mechanism by which you get alternation between one animal and the other animal. This is also corroborated by a really beautiful recent study by Benichov and Valentine that was published this year, in which they looked at turn taking in the finches and where only the males sing. And in this case, they're actually not looking at song, they're producing these vocalizations from the male that I in my head think of them as saying, hey, 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 hey. But when they do that, it's actually an alternation between one and the other one. They take turns. And so they had this experiment where they had the male saying hey and the other male saying hey back as a playback. And they found that the live male would actually alternate his syllables with the playback male. And then they found that if they applied mucimol to HPC, a GABAergic antagonist, that would that they lost this ability to take turns, suggesting that it's a GABAergic circuitry with an HPC that's responsible for turn taking, at least in finches. So what we'd like to go do now is kind of repeat the same type of experiments in these wrens and see if this GABAergic circuitry is important for turn taking in these animals. We want to do those experiments in the future. What we did do, though, is look at the effect of, we wanted to see if we could see this auditory input to HPC. And so we did the same, so we took the birds after they sang and anesthetized them with urethane to see if we could uncover these auditory responses from the partner bird. And in fact, it's been shown, urethane is a complicated drug, but it has been shown that urethane inhibits GABAergic transmission in vertebrate animals. And so what we did after the birds had sung duets, we anesthetized them with urethane and then played back the songs that they had sung to look at the sensory responses in HPC. And I'll show you one of those experiments here. And so in this experiment, this is the data I've been showing you previously, where the animals sang this song. And you see nice alternation of activity between the male and the female, and potentially an inhibition of activity in the receiver. We then took these birds, took off their chronic electrodes that were on their head after we anesthetized them with urethane, and then played back this very song that they sang earlier to them. And here's the responses that we found. And so in this case, here's under urethane playback. Each of these dots is response in HPC when we played back the song, and we played back the song 20 or 30 times. So we get these raster plots where each line is a playback of that song in the male and the female. And then we created a histogram with those, which are shown here. And what we find is what we reported in our previous study, is that when the male sings, we now see an increased activity in HPC response. When the male sings, we see an increased activity in HPC in the female. And when the female sings, we see an increased activity in the male. And that's quantified in this next slide. And so here, we see that if we look in the brains of the receiver, in urethane anesthetized animals, we see the HPC is excited in the receiver. And just to remind you, this is in contrast to what we saw in the brains in HPC when the animals are awake, where we saw an inhibition of activity. And so we think what's happening is that in the awake animal, this auditory input is gated, and you get the auditory response in awake animals. The auditory response from the sender in the awake animals activates a GABAergic circuitry that inhibits responses in HPC and prevents HPC from being active, which allows this nice alternation between the two animals. And that's just illustrated here on this next slide. We come back to the schematic where we have two individuals, the male singing his part and the female singing her part. When the male sings his part, he produces a syllable here. That syllable is sent as an auditory signal to the female. And that auditory signal gets to the female and activates GABAergic circuitry in the motor system in HPC and alters the patterning of the CPG within HPC. It alters the timing of her syllable. When she sings her syllable, that is the auditory signal that gets sent to the male. That auditory signal goes to the male and activates GABAergic circuitry in the male HPC that alters the timing of the production of his syllable. And this produces this very nice alternation between the two individuals. Of course, we would like to go back and more directly manipulate the system by manipulating the GABAergic circuitry, but that's the next set of experiments. Okay, so we go back to the original question I asked was, what happens in the brain of the receiver? And the prediction was that there would be excitatory auditory responses in HPC of the receiver. Well, in fact, there weren't excitatory, but there were inhibitory auditory responses in the HPC of the receiver. And so we were kind of correct. And so I'll give us a very small check mark for that. Okay, so when we're looking at what happens in the brains of the senders and the receivers that help coordinate the activity between two individuals, we found that there was an increase. There's, of course, a motor response, which you would predict because the animal has to produce its own behavior. So we saw a motor response in the brains of each individual, and those alternate. And they alternate because the auditory input from one inhibits premotor activity in the receiver. And this is the one thing that actually now provides this really nice alternation between the two individuals. Okay, so just to kind of summarize, what I've shown you is we were interested in how these two animals produces very tight coordination. Behaviorally, we found that it's the auditory feedback from one animal to the other animal that's important for the timing between the two individuals. And when the male sings, he produces his song that goes to the brain of the female that influences, that sends a signal to the motor system of the female. She produces her syllable, and then that gets sent to the brain of the male that changes the timing of his CPG, and you get this nice alternation back and forth. Okay, and so we started with this question of how the nervous system coordinates activity between two individuals. And so in this example, we have turn taking in these two animals, these two wrens, where one animal produces a signal, communicates to the second animal, which then responds and produces a signal and communicates back to the first animal. And so what's interesting here is that we see that how you coordinate the brains of these two individuals is something that's not surprising, but it's really the sensory signals that's shared between the two that really links the two brains. And so here, for example, in the blue-footed booby, the female's receiving the input from the male visually and will respond. And then in this case, there's a visual interaction between these two that tells something about the social interactions between these individuals. And importantly, in vocal communication, one individual is saying something and the other individual has to listen, which I think is a very good lesson for all of us at this time. And with that, I will finish. And thank you to everyone that's helped with these experiments, particularly the wrens, but also my longtime collaboration with Eric Fortune and some of his students, Pamela and Andrea. Of course, our electrophysiology guru at Nancy Day, who's now at Whitman College. And we also like to thank Stephanie White, who loaned Nancy to us when she was a postdoc in her lab. Students from the Claremont colleges, Jose, who helped us at the Yanayaku Biological Field Station, other people in Ecuador who's helped us collect the data, and Ignacio Moore, who's at Virginia Tech, who loaned us his truck so we could take all of our equipment up to the field station, which is harder than it seems. And then we also want to thank a big shout out to the National Science Foundation to providing the funding for this. And finally, I'd like to say that in my department, we are hiring. The ad will go out soon, I hope. We want a neuroscientist using computational tools who can engage undergraduate students in research and can teach a computational neuroscience course, broadly defined, and contribute to our neuroscience curriculum. And then if you have any questions, please contact me, and I'm happy to answer questions about that. Okay, and with that, I will say thank you very much and take some questions.
Yay! Well, as people are figuring out how to use the ask a question function, which always has a little lag, I have, of course, like 1,000 questions. One thing is I'm curious, do you have guesses from the anatomy or an interest in figuring out where the inhibitory input is coming from? Yeah,
that's a great question. I mean, you you mentioned is the intro that there's the thalamic nucleus that provides input, and wouldn't it be great if it was like came full circle that was the case? So that's definitely a possibility, right? That you have auditory input coming from the thalamic nucleus that then provides this signal in. I think actually it's an excitatory signal that's activating the inhibitory circuitry within HVC itself. How it's working, but I don't know. I think if I were to do that, my first guess would be the thalamic nucleus I studied in Rich's lab, UVA. I'm not going to do that in these birds because it's really deep and really small and hard to get. So this is not the species to look at that in, but that would be my guess.
Cool. Yeah, I have one more question. So there's some recent work like from Dmitry Arnov showing that like the bird hippocampus, which is actually at the surface, since we're talking about easy to record places, has some roles in like sequence generation and maintenance. And sequences are something like you hear all the time in hippocampus talks. This kind of looks like a sequence to me. I was just wondering if you had thought at all about maybe looking at the easy to access bird hippocampus.
I have not. It's a great idea. We've focused on, we're so centric to bird song. And of course, I come from the background of zebra finches where everything's, in the song system with HVC, and it's been shown to be a pattern generating circuit, which is also why we looked there. But it's a great idea to actually look at hippocampal things. I don't even think in birdsong people looked at hippocampal connection, physiological connections to HVC. It seems like someone's done that. It's nothing that I unfortunately have know very well. So that's a great idea. Yeah, I like it.
Okay, so we have a question from Bradley. How flexible is the timing of song alternation? Would you expect successful duetting in an artificial playback system that had arbitrary timing? And do you know anything about kind of the development? So do juvenile birds learn from duetting adults or from single adults?
Yeah, these are sort of the $1 million questions or $1 billion questions that we'd like to know. So the first one is how flexible is your timing? What's interesting about these birds, it's different in other species of birds that duet. In these birds, we can't get them to alternate with a fake, playback. So we can't get them to match. So if we go up and play a duet to them, they'll match that duet sometimes, which is interesting. But they do it in a duet. So both birds come forward and they both duet over this other duet. If we play a song where it's like a solo song, so the female singing with the gaps, they won't engage in that. And so the only way we can actually get them to duet is to have two individuals live. They're pretty flexible though, because we know when we've been out walking in the field and we'll play a song or we just hear them, they can be up to 30 meters apart, and they sing very loud. I didn't say that. They sing about 80 decibels. If you're next to them, it actually hurts your ears. So this gets through the dense bamboo, which is great. And then they come together, and we think why they come together is not to see each other, but because it helps with their coordination. But there's a lot of flexibility in that. And that would be a really cool thing to do, kind of what they do, in Eve's lab and others' labs is when does the system fail, right? And so how far apart do you have to get before the system kind of fails? What's the limit of that? And we haven't done that. It's really hard in these animals because of they're so kind of robust in what they do. The second question, which I would love to know the answer to, is how they learn the song. We don't know. In addition to living in dense bamboo, they set up these nests that I've only seen one of them, and then you get these torrential rains that come through, and all the bamboo falls down, and so they have to move. So it's really hard to find nests, and it'd be really difficult to do the experiment. We do know that in addition to singing these duets, which is amazing, they also sing in choruses. And so you get groups of animals singing together. And we think that these choruses are produced by family groups. And so maybe that's one of the ways that the animals actually learn, right? I'm a female bird, and so I'll sing with my mother to learn my parts. and the male sings with his father to learn his parts. Let's see, I had another point. So we don't really know. Do they learn both parts and then know that they're a male bird or a female bird and then sing their part? There's one study, it was never produced, but I talked to Rachel Levin, who did this in a bay wren from Panama, and it's a very similar song. And she actually raised these birds. She took them back to the lab and raised them in the lab. And what she found was that the birds sing to, if I remember correctly, the birds sing to learn the parts of each individual and then know that they're female and then produce, for example, know that they're female and produce the female parts. So we don't know, but that's really something that is really critical to understand. Yeah.
That's very cool. We have another question from Annie. When humans do conversational turn taking, there's good evidence they predict the end of their partner's utterance, which is why turn gaps are so short, about 300 milliseconds. Any evidence that wrens actually predict the timing of the partner's syllables versus being like solely inhibited by the actual singing?
Yes, I think they do. That's really hard to pull out from the data, but we have some data to suggest that timing between when they hear and when they respond, particularly when we start to put these delays in, is actually shorter than you would predict it would be based on the delays, which suggests that they really are predicting what's next. So, and I think that's also part of this pattern generating circuit, right? So the pattern's already there, you're just changing the timing. And so the prediction is actually not that they're kind of mind reading the other one, but it's the auditory input that's affecting basically the intersyllable interval, because that's already determined by this CPG that's in the brain of the one animal, if that kind of makes sense.
Very cool. How stereotyped are the songs like across pairs?
Oh,
and, like, can you swap a male basically or a female?
Yeah, so these birds are even more remarkable because what I showed you, it suggests that in finches, they sort of sing this one song. And what I showed you, it almost suggests that they sing something that's very similar across different renditions. But when we go in and actually look at the duets that are being produced, we call them jazz singers because there's a lot of variability in each of the syllables they produce. And so when we go in and we tried to sort of match syllables across renditions, it was almost impossible to do so. But there's some sort of code that they live by, because we were making up some songs to play back to the animals, and we swapped out, if the male sings CE, we made it EC, and it sounded wrong to us, even my ears. So there's some sort of code that's there, but I'm not smart enough. I'm not as smart as a wren to figure out what that is. But we can, another part of what your question is, the other thing we've done is caught birds in different locations and brought them back together, and they've learned to sing with each other. And so they're actually bad at it to begin with, and they overlap each other a little bit, and their songs are really short. But as they spend more time together, their songs get longer and longer. And in fact, we sort of know when it's a mated pair because their songs can be up to like 30 seconds or two minutes long. And that seems to occur over time. And probably part of the function of the duet to show that they're a mated pair and are like really good at what they do, basically.
There's one more question in the chat. This is following up on the last, 2 questions ago now. Do differences in timing correlate with a partner not singing or dropping out transiently, as in the example?
Can you repeat that? Sorry.
Yeah, I see. Do all the differences that you see in timing tend to correlate with a partner kind of forgetting something or not hitting their syllable in the duetting? Like, do you ever see these like changes in timing that are not like directly related to the partner not like messing up, basically?
No, let's see. I don't know how to answer that. So when we do see, it's mostly the male we see dropping out, and then you'll see the female adjusting her syllables. We do, when they sing solo song, we do see that the intersyllable interval changes, and that's when the female and the male. And so we do see kind of those changes. We don't really see, I'm trying to think. We have seen some overlap sometimes, maybe as part of your question. So do they actually ever overlap? And we do see that sometimes, and that seems to be in these pairs where it's happened a couple times. One is we've caught pairs in different places and brought them together, and they're not good at coordinating. They'll overlap a little bit or kind of be sloppy and then get better over time. Interestingly, and sometimes when we've done these distance experiments, we put them at 7-10 meters apart, and then we bring them back together, then they'll actually overlap some, suggesting that they are kind of anticipating the timing of the other individual. Maybe that kind of gets a little bit to that question.
Yeah. Well, this has been awesome. Thanks so much for joining us and closing out 2020 season of this whole series. It's been lovely hearing from you today, and thanks everyone for coming and asking questions. That's it.
Okay. Thanks everyone for your attention. I really appreciate it.
Bye.
Bye.